False Positives and False Negatives webinar. My name is Karen Whitman, and I will be the moderator for today's webinar. Before we begin the presentation, I have a few housekeeping items to go over. The webinar is recorded and will be available in 1 to 2 days. The questions will be answered at the end of the presentation portion, so any questions in the queue that are not answered during the presentation, Doug will follow up with after the webinar. When questions do arise, go ahead and enter them into the queue by clicking on the questions section of the GoToWebinar panel and simply typing your questions in. It is now my pleasure to provide you some background on today's presenter, Dr. Doug Marshall. Dr. Marshall is Chief Scientific Officer with Eurofins Microbiology Laboratories, Inc., a division of the global life sciences company Eurofins Scientific. He also is co-founder and director of Food Safety Institute, LLC, and an integrated consulting and analytical services company affiliated with Eurofins. He currently holds adjunct professor positions with Colorado State University and Florida State College. With over 250 publications and over 150 invited presentations, his scientific research and outreach interests focus on improving microbiological quality and safety of foods. Dr. Marshall, the floor is yours.
Thank you, Karen, and good morning, everyone. I really do value your time. We're going to run through this pretty quickly. Obviously, the title was attractive for those of you in attendance, and we're going to run through some of the things that worry me about food microbiology testing, and then I'll introduce some of the things that laboratories can do to reduce my worry works. So for those of you unfamiliar with Eurofins, just a quick introductory slide. Most of the markets that we participate in food testing, environmental testing, and pharmaceutical testing, we are the #1 market shareholder in those locations. And we have a very simple mission in our company, that's to contribute to global health and safety by providing our customers with high-quality laboratory and advisory services. Some of the things that contribute to false positives and false negatives are the following. And so when you are contemplating whether you're running an in-house lab or outsourcing to a third party like Eurofins, these are some questions I think are deserving of your time to contemplate. So first and foremost, is a laboratory accredited to perform tests? Many laboratories will claim they hold ISO 17025 accreditation, but what you really need to dig down on is what tests within their portfolio are actually in the scope of that ISO accreditation. So for example, you could be ISO accredited and the only test on your scope could be anaerobic plate count. So all the other things that might be supported in that laboratory may not be on their scope. Second question is, can laboratory personnel describe and perform the methods? Is a laboratory capable of performing sample preparation? So as a large global laboratory network, we test almost every kind of specimen you can imagine. And one of the real issues is just what do you do with that sample on presentation at the benchtop? Some items are very, very challenging to break down, to be able to do very good analyses. Other items like liquids are easy to use. Other compositional issues associated with those matrices also are important. Are they very high pH, very low pH? So there is an opportunity for doing some sample manipulation to increase the odds you're actually finding and counting the microbes you're interested in. Also, another question you should consider is the methods are being deployed in the laboratory. Do they have some sort of independent third-party approval? So it might be very nice if I have an in-house test, but if it hasn't been thoroughly validated, you probably should question whether or not the results are actually good or not good. Another item is, are the methods validated for the matrices of interest? So make sure that the method that you're using is fit for purpose. And then any non-standard methods needs to be justified and validated for your particular matrices and for the analytes of interest. So any issues on this particular page that are not fully resolved to your satisfaction have the potential to increase the odds of having false positive and false negative test results. There is some method guidance available from U.S. regulatory agencies. These are the URLs, and you'll have access to this webinar deck afterwards if you click through these. But these are very good just in terms of providing advice to deploying appropriate microbiology methods. When we look at methods for inclusion in our portfolio, what we're really looking for are the validation studies associated with those methods. So some terms that you might see when you're evaluating methods, I'll run through on the next couple of pages. Inclusivity is the ability of a method to correctly detect a large number of target strains and not detect a large number of other potentially competing microorganisms in a similar matrix. Exclusivity is a term that describes that there is no observed interference from non-target strains in the test. Specificity is the ability of a method to correctly detect a pathogen of concern and not give a positive reaction to other microorganisms that are not the target strain, species, or genus in the sample. So, for example, a test that has 100% specificity will give you no false positives observed while all contaminated products are detected. Obviously, that would be an ideal test. Sensitivity relates to the limited detection or the limited quantification of the method. So in most circumstances, particularly when we're talking about pathogen detections, we want a very, very low sensitivity level. For example, the ability to detect low populations of pathogens. So for example, 1 colony forming unit per environmental smog. A couple of other terms you need to understand is a presumptive test result means that the test platform provided a positive result from screening a sample enrichment. So a presumptive may or may not be the actual organism you're interested in. All we know is that that test result gives us a presumptive positive result. Further testing is usually required to be able to get a confirmed result. So in this case, what we're doing is we're taking the sample enrichment and we're doing further isolation and plating to be able to actually find a viable pathogen, and we use biochemical testing or molecular methods to confirm the presence of the pathogen. So in many circumstances, our customers only react when there's a presumptive positive because they don't want to wait another potentially several days to get a confirmed result. So what then are false positives and false negatives, and why are they so evil? Well, let's talk about the reality. So we have two potential scenarios when we're doing a pathogen screen. The first is the pathogen is present. The second is the pathogen is absent. There is no almost pathogen there or almost pathogen absent. It's either present or it's absent in the specimen that is presented to the laboratory. When you look at the analytical method results, if that result is positive and the pathogen is present, then that result is a correct result. However, if the method result is positive and the pathogen is actually absent from the specimen, then that's what's called a false positive result. If the method result is negative and the pathogen is present, then that's considered a false negative result. If the result is negative and the pathogen is absent, then that is a correct result. So what we're really dealing with here are false positives. This is when a pathogen-free product is actually rejected because of your test result. Compared to a false negative is when pathogen-containing product is shipped. And so what we don't want to be in a situation is to have a food manufacturer exist in an environment like this. They've got their production lots, they get their test result back, and there is always going to be the potential for doubt whether that test result is false positive, false negative, or a true test result. I'll show you some data that describes the extent of false positives and false negatives that the laboratory testing industry is experiencing. This is a study that was done by the American Proficiency Institute amongst their laboratory participants in the proficiency test programs. They surveyed over a 14-year period their test results in their PT program for E. coli 157, Salmonella, Listeria monocytogenes, Campylobacter. And what they found over that 14-year period is the false positive rate, if you look at the first column, ranges between about 2.5% up to 4%, and the average over that 14-year period is about 3%. So you might ask the question, well, over that 14-year period, we've had an improvement in methods, greater sensitivity, greater selectivity. You would think that the rate of false positives would have declined. But if you look at the most recent data that they had in the study was 2012, that false positive rate on average remained at about 3%. If you look on the third column to the right, the false negative rate of those tests, on average over that 14-year period, was about 6.6%. So again, the low was 5% and the high was 9%. So when you consider a false negative rate, that has pretty significant implications to the customers because companies are shipping a lot of contaminated product. More recent data, we've seen an improvement in those results. The average false positive rate has declined precipitously by about half, down to 3.4%. And when you look at the dramatic decrease in the false negative rate for Campylobacter, that is quite a stunning improvement in the methods. So these test data then amounted to about 40,000 different data points. So it's a pretty robust data set. And that significant decline in false negatives obviously is very good for improving public health. So what then are some of the evils of false positives? Why do the laboratories take this very seriously, and what are the implications to the food manufacturers who are our customers? Well, these are some of the potential consequences. So you have unnecessary product withheld or recalled. You've got costly rework or disposal. You know, costly break in production schedules to try and figure out why we had a positive test result. You've got expenses associated with line cleanup. Obviously, if you're not shipping product, you're losing sales revenue. If you report this as a reportable food registry event, then you have the risk of damaging your brand reputation. This could also lead to increased regulatory scrutiny, increased auditor scrutiny, and an unnecessary reassessment of your food safety programs, and obviously loss of trust in your laboratory service provider, whether it's your own lab or an outsourced lab. What are some of the potential causes of false positives? This is a list of some of those. First one here is you could potentially have a large population of non-viable cells. These may give detection, but are not confirmed due to the inability of the pathogen to grow after enrichment. So again, you have a presumptive positive, but difficulty in confirmation. Second potential cause is you could have related microorganisms that are detected by a platform. So for example, in some platforms, Citrobacter is a common cause of false positive salmonella because the antigens that are present on the bacterial surface have molecular similarity to the antigens that salmonella presents. So again, your platform's detecting salmonella, but it's not there. You're detecting something else that's closely related to salmonella. You can have a large population of competitors and enrichments that prevents your ability to pull a pure culture pathogen from those enrichments, which then would lead to confirmation failure. So in this case, the pathogen is actually there, but you're not able to confirm it due to a large population of competitors. You could potentially have cross-contamination during sample collection, transport, and laboratory preparation. So this cross-contamination could occur when the individual and/or individuals are collecting the specimen at the production sites. If the containers are improperly sealed and you get cross-contamination within your shipping container, that could be a potential cause. And then sloppy aseptic technique in the laboratory could also introduce the organism to the specimen, even though it wasn't originally present in the specimen. Likewise, in the laboratory, you can have cross-contamination with the amplicon when you're running PCR assays. So in this case, during the sample preparation, to present a sample to your PCR platform, if there is organism DNA in that environment and it gets into that preparation, then you can get a false positive result. One of the interesting things about false positives is what's known as a false positive paradox. What happens here is when the pathogen prevalence rate is very, very low, and you would expect that to be the case in most processed foods that have a kill step, for example. And that rate is so low that it's actually lower than the method false positive rate. So in this case, the method false positive rate is greater than the prevalence rate. So this is a potential problem when finished product testing, when you have perfectly good food is withdrawn based on test results. So let me give you an example. Assuming that a pathogen prevalence rate of 0.01%. So what this equates to is 1 positive per 10,000 analytical units. Most of you producing finished products with a kill step would be very unhappy with this positive rate, but it's potentially observable. Let's assume that you've got an outstanding method. Remember, the methods are going to give you a false positive rate of about 3%. So in this case, we're using an exceptionally sensitive method, but that is 0.05%. What that means is it's 99.95% accurate, an exceptional method. What this means is you will actually reject 5 units out of that 10,000 based on just the false positive rate of the method. So what this gives you is an 80% greater rejection rate than necessary. That has huge economic implications for food manufacturers. I want to do another calculation study for you to think about. So what does a 3% false positive rate mean? We make the assumptions that the average American eats about 900 kilograms of food per year, and in the U.S. there's about 320 million of us, and the U.S. produces greater than 285 billion kilograms of food annually. Then a 3% false positive rate would mean that we are withdrawing 8.5 billion kilograms of food due to this pathogen test that has a 3% false positive rate. So what that means is that 8.5 billion kilograms is going to have to be recalled, disposed, diverted, or reworked, which adds extra cost to food manufacturing. What you also have to understand, if you're disposing of this product, that's enough food to provide a yearly ration for almost 100 million people. What are the practical implications then of false positives? Well, again, a detection platform gives a presumptive detection. Your subsequent confirmation steps fail to isolate a culture or to confirm. What do you do as a food manufacturer? Do you go ahead, so some of the things you're going to ask is, did the sample really contain the pathogen? And if it did, the assumption is that a production lot from which that sample was withdrawn is contaminated. So how do you know what caused the confirmation failure? Do you blame the laboratory? How can the laboratory prove its innocence? And is it wise to actually ship product when you have a presumptive detection but no confirmation? Let's turn now and start talking about the evils of false negatives. So in this case, what the food manufacturer is going to be doing is shipping contaminated product because the test result comes back negative. And so the assumption is that sample was representative of the lot. If the sample came back negative, the production lot is considered to be negative and therefore ships. If that happens, then obviously you have a risk of injuring or killing your customer, which is not a long-term successful marketing strategy. You are playing Russian roulette, but you won't be caught. So again, I know some food manufacturers out there that prefer to use very insensitive tests because they want to reduce the odds they'll find something. You run the risk of losing your customer base with either a recall or an outbreak investigation. This can lead to damaged brand reputation, increased regulatory scrutiny, increased auditor scrutiny, and your entire food safety program, you would have no indication that there is an issue somewhere in that program. Obviously, if you're using a laboratory provider for doing these tests, you're going to lose trust in your laboratory provider with a false negative. So what are some of the implications? Let's do another little back of an envelope calculation for you here. So let's make the following assumptions. So if you have a 3% false negative rate, that means 3 out of every 100 meals is contaminated. So it's just an average meal from an average consumer like you and me. It could also mean that 3 out of every 100 packages will contain a pathogen. And you would never know because of your false negative rate. So over a 100-day period, you should be sick 3 times due to a foodborne illness, if this is really a true reflection. So CDC estimates the actual rate is about 20 to 30% of us will get a foodborne disease this year. So that's a bit of an over-assumption on the number of illnesses associated with this product due to a false negative. Let's make another assumption. The average person eats a little under 5 pounds of food per day. And that is about 6 times what a typical 375-gram sample would be. And so again, this is a very large sample size for doing salmonella screening. So if you have a 3% false positive rate, go down here on the bottom calculation, times 2,300 kilograms, divide that by 6, this assumes that you're eating salmonella in finished products about every 11 days. So the first calculation and the bottom calculation, that's a pretty stunning exposure rate to salmonella. There could potentially be out there people consuming product that actually has salmonella, but the test result comes back negative. So what are some of the causes of false negatives? Well, at very, very low prevalence rates and population sizes, there are small odds of finding a pathogen in a pulled sample. So again, it's just random dumb luck that you'll actually find it, even though the production lot is contaminated. The methods assume that the pathogen of interest is uniformly distributed in the specimen. That is rarely the case. So you are going to have non-homogeneous distribution and potential hotspots. You could have overgrowth of enrichments by competitors that prevent detection or recovery of pathogen. And you could have what are the so-called viable but non-culturable cells. Some people call these dead cells. These may not give detection or grow during confirmation steps, but could cause illness if they are indeed viable. If they're dead, they're not going to cause illness. You could have a sample from a food specimen that contains antimicrobial agents that are activated during the enrichment process that could prevent growth or kill the pathogen. So again, even though the pathogen could be present in the specimen, you're not able to detect it during the test process. And you also have the potential of having very, very slow-growing strains that you're not going to be able to do confirmation with because you can't pull them out of those enrichments. So again, the methods assume uniform prevalence. On this particular side, each dash is one, let's say, Listeria monocytogenes cell in a large production lot. So each square then is a potential grab sample for you that you can either pull and send to the lab, or pull multiple sampling units, composite those, and send to the lab. So in this case, this production lot obviously has Listeria, but not every lot is contaminated. But the Listeria is uniformly distributed in that production lot. So even though the lot is clearly contaminated, there are many vacant sampling units here that if you pull that, you're still not going to find Listeria, even though the lot is contaminated. Let's assume that you have an exquisitely low prevalence rate of the pathogen in your production lot. So in this case, you've got one analytical unit that has Listeria monocytogenes, but all the remaining possible sampling units are free of that organism. So again, you can see you have exceptionally low odds of finding that at that low prevalence rate. And in fact, when you have this kind of prevalence rate, if you talk to the regulators, they will say, you need to be using a statistically valid sampling plan. Well, in this particular instance, you would need to sample about 95% of these sampling units to be statistically valid. That means you're only really able to ship 5% of what you produce. So that's an unreasonable expectation from the regulatory perspective to have statistically valid sampling plans at extremely low prevalence rates for finished products. You also have poor odds due to non-uniformity. Here's an example where you have a fairly high population of pathogen in your production lot, but it's not uniformly distributed. In this case, you only have 2 sampling units that have the pathogen present. All the other ones are negative. So again, the odds of you finding this production lot that's contaminated just by pulling 1 or say 5 samples is very, very low. So how do you increase the odds? Well, in FDA BAM, in their Salmonella section, they talk about a risk-based sampling plan that looks like this. So for Category 2 foods, these are going to be foods that the customer is actually going to cook prior to consumption. So the customer, the consumer, will apply a kill step. Therefore, the belief is that these are about as low-risk products as you can, because at the point of consumption, you'll have a kill step applied. So in this case, what FDA recommends is you pull 15 25-gram analytical samples, or you can pull a 375-gram representative sample. Category foods are those that will not have a kill step applied prior to consumption. Therefore, those are considered to be ready to eat and potentially higher risk products. In that case, FDA recommends you pull 30 25-gram samples, or you could pull a single 750-gram sample. A Category 1 food is a food that's ready to eat and destined for a high-risk consumer. So in this case, it could be food going to hospitals, to daycare centers, and the like. In this case, FDA recommends pulling 60 25-gram samples, or pulling a single 1.5 kilogram sample. So those of you who work in a laboratory, imagine having to enrich a 1.5 kilogram sample. So no one does that just because of the size. So what you're doing is you're breaking this down into 4 375-gram samples and enriching 4 samples and looking at those combined test results. So, you could take this to whatever extreme you want. The more samples you pull, the greater odds you'll find that non-uniformly distributed, very low prevalence rate pathogen in your production lot. So, I want to kind of finish up here and talk about some things that lead to false negatives. First example I'll give you is we see this occasionally with some of our customers. They'll run a total interbacteriaceae count, and if the test result comes back less than the limit of detection, so in this case it would be less than 10 CFU per gram, then the customer assumes the absence of Salmonella because Salmonella is a member of the interbacteriaceae family. Well, there's a couple of issues with that. Number one, your limited detection is quite high. And number two, Enterobacteriaceae may or may not be correlated with the presence of salmonella. So unless you've done that, built that correlation for your products in your environment, this is not a good assumption to make. Improper sample preparation before running an assay is something that is common. And we see this frequently with foods that have antimicrobials, whether added or natural, as part of the product formulation. And during that enrichment process, you're basically bathing your pathogen in the presence of those antimicrobials, consequently leading to inability to detect or confirm. When you're doing these very large 375 gram or 750 gram enrichments, you're adding your specimen to your enrichment broth, and that enrichment broth is not pre-warmed. You stick that room temperature enrichment broth in an incubator, it's going to take many, many, many hours to get to proper incubation temperature. During that time, your target of interest may not be multiplying and may not get to the detection limit of your detection platform. Hence, the organism's there, but you're going to get a negative test result. Sometimes product actually will float on the surface of these enrichments. So unless you get that product fully immersed in that enrichment, you're not likely exposing that product to a culture condition that will allow multiplication of the pathogen. Another item we see is failure to add enrichments, selective agents that sometimes are added to these enrichments to reduce the potential for interference from competing microorganisms. Another common failure rate is failure to incubate enrichments long enough. Industry hounds us every day. We need results faster than you're able to receive. Well, the problem is we can't change biology of the organisms. So there are platform providers and laboratory service providers that will claim they've got a reduced turnaround time on their test results. I would be skeptical because what they're doing is they're giving you what the customer wants, but it may not necessarily be a very good test result. Because again, if the organism is not growing to a population that can be detected, your test result is going to be negative. Every once in a while, will we see poor quality detection and confirmation platform reagents. So in other words, the detection platform just isn't working. The only way to detect that is you have to run positive control. So if the positive control fails, that tells you there's something wrong with the method. And one of those failures might be that your detection wells and your detection platform have faulty reagents. We also see a failure to pick atypical colonies that may be pathogens during that confirmation process. So again, a lot of these confirmation platforms are using visual appearance of colonies. And every once in a while, you'll have a particular strain that does not give you a typical appearance. So a laboratory technician might overlook that atypical colony and go ahead and call the result negative, even though the pathogen is present. A couple of additional items is there's a failure to isolate pure culture for subsequent confirmation. So in this case, again, the detection platform gave you a positive, but you're not able to confirm. So you have to ask yourself a question. Was the pathogen really present in the sample or wasn't it? Sometimes we see the inability to do serotype confirmations due to poor quality sera. And of course, human error is ever present in anything that involves human activity, and laboratories aren't perfect. So we can have wrong calls by laboratory technicians on detection confirmations. And one of the things we do in our laboratories, we're not using test platforms that require that kind of subjectivity. Let's talk about some of the bigger picture issues here. This is a nice slide I got from an old publication by Todd Kleinhammer's group that kind of shows if you're a microorganism and you have to exist on the left side of your screen in a product or a carrier, so it could be a product specimen or an environmental sample, you are exposed to all kinds of environmental insults that make life rather difficult. So you've got water activity pressures at low water activity. You've got the potential for heat shock, heat damage, chill shock, slow growth at cold temperatures. You've got competing microorganisms. You've got a packaging environment that may not be conducive for your survival, such as packaging or high carbon dioxide, modified atmosphere packaging. And then you've got the potential presence of either natural or added antimicrobials. So if a food manufacturer is doing everything correctly, all of these barriers that are applied during processing are quite insulting to a microbe. On the other hand, once that microbe gets presented to a customer, then it has the following insults in the body. So you've got bile and mucin and proteases that are part of your natural digestive tract. You've got suboxygen, but usually at limited supplies. You've got competitors that could produce bacteriocins. You've got a exposure temperature that may not be optimal for your growth. And then you've got a really low acid environment in your stomach, and then then an alkaline environment in your intestinal tract. So again, these microbes have lots of insults to be able to survive in foods and during digestion. So because of that, the strains that you present to the laboratory may not be very healthy strains. They might be injured. They might be not able to grow at optimal conditions. I picked a few examples here of some detection platforms and the recommended incubation times of those platforms. And VITUS is an ELISA-based kind of detection platform. PAX and MDS are both a couple of common PCR platforms. And these are the incubation ranges for Salmonella. So if you look down there, at the extreme end, you're looking at 24 hours to run a full enrichment process for most of these methods. At the short end, you're talking about 16 hours, in one case 10 hours for lettuce on BACs. And so my concern with some of these is, what is the laboratory doing with your samples during this enrichment process? The laboratory's got discretion on when to pull samples for detection. So I want to give you an example of a typical laboratory workday. Let's make the assumption that samples are received at 10 o'clock in the morning. Samples that then get processed, registered, the methods are selected, and so forth. So that takes a little, give the laboratory 2 hours to make that happen. And the samples get in the laboratory, and they're set at noon. You're collecting samples. You put them in the incubator at 2 o'clock. So the incubation period is going to start at 2 o'clock in the afternoon. So if you're going to enrich those samples at 24 hours, the earliest read the laboratory is going to get is 2 p.m. the next day. It's going to take a few hours then to do quality control, to prepare the analytical report, certificate of analysis, and so forth. So it's likely you'll get those results later in that day. Depends on the lab, depends on the workload, and so forth. Now, then imagine you're a manufacturing laboratory says, I can go ahead and give you a test result several hours earlier if I use the low end of that enrichment time, let's say 16 hours. So in this case, the earliest read is going to be at 8 a.m. the next day. So you could potentially get that test result, go ahead and start production for that day, thinking that everything is all is well and good. Let's also assume that a laboratory really wants to make you happy, and they're going to choose a 10-hour enrichment. The earliest read you're going to get is going to be midnight the same day, so before you show up to work that morning, you're going to have your analytical result. So my question to you is, let's assume if you've got a negative test result at 10 hours, what happens in those enrichments between 10 hours and 24 hours of additional incubation? And what is the risk that the pathogen could not be detected at 10 hours due to its growing very slowly, but it could be detected at 24? How many false negative test results are you going to be receiving if you're shortening the enrichment time? And that leads me to conflict of interest. So in this case, does the company management have a conflict of interest in their testing programs, and are there protocols in place to mitigate such interest? So for example, are laboratory employees trained in ethical behavior regarding proper sample collection, testing, and reporting? Are there written non-confirmation policies? Are there undue influences that impact the integrity of test data? And are methods used that are fit for purpose? So again, those of us who run laboratories, we have lots and lots of different platform providers. Some of these make outlandish claims at their ability to be able to give you test results. You know, I've seen things in 6 minutes, in 30 minutes, in 3 hours, and so forth. And are those methods really fit purpose? If you want to buy the cheapest, fastest test result you can get, that's always going to give you negatives, that's a conflict of interest. So in conclusion, things you can do to reduce your false positive and false negative rates are the following. Make sure you're using validated methods. Have those methods been validated for your analyte and your product? Is your lab actually functioning correctly? Our negative test results, they do not imply that the lot is negative. All that does is it says that the sample that was presented to the lab did not have the pathogen using the detection platform that the laboratory was using. So this obviously means that you've got implication for test and release programs. You really need to be making sure that all of your food safety programs are working well, because that end product test result is more of a way to detect gross production failures rather than being a good way to detect negative lots. And then lastly, what do you do with a positive test result? Again, it may not be confirmed, it's just a presumptive positive. Do you ship product and wait for confirmation, and how much faith do you have that that confirmation process is really actually able to find the pathogen if it has been presumptively detected? So I thank you very much for your interest and time in this webinar, and we do have several minutes available for questions, and I would be happy to address those.
Thank you, Dr. Marshall, for sharing your insight on false positives and false negatives. We're now going to move on to the question and answer portion of this webinar. So everyone, please go ahead and enter your questions into the queue. We're going to start off the questions and answers with this question for you, Dr. Marshall. What are examples of antimicrobial agents that are activated in enrichment?
Okay, it could be almost anything. What we particularly see with that scenario is you're talking about a dry food product that could have, let's say, antimicrobial containing spices. This could be things like cinnamon, cloves, onions, garlic, and so forth. When you hydrate that food product, then you are adding water that acts as a solvent that can take those natural antimicrobials and present those to any pathogens that could be present, or even other microorganisms. Let's say you're doing quantitative microbiology. And those have the potential to slow the growth rate, or in some cases actually kill the cells. So that would be the primary example that we see. Other examples could be that you've got ingredients that are present in the formulation that, again, when you do that enrichment, if those ingredients are added to give you extended shelf life to prevent microbial growth, then they're also going to be potentially effective even when diluted in that enrichment. Okay, next, please.
The next question is, there are many vendors selling pathogen detection platforms that promise fast turnaround time. Do they have issues with false negatives because of short incubation times?
That's a question you're going to have to ask those vendors, and you're actually going to have to challenge that platform with your products for the pathogen of interest to be able to answer that question. So I would just ask you, like most things, if it sounds too good to be true, what is your validation data that says it's actually true to your application? Make sure that that platform is fit for purpose.
Okay, thank you. The next question in the queue is, if the lab is not able to confirm salmonella after a presumptive positive test results a finished product, is it okay for me to ship?
I can't answer that question because that's a risk that the manufacturer needs to bear. Obviously, there is potential risk. You have to ask yourself a question. The pathogen detection platform that was being used on your specimen by that laboratory came back positive, but the laboratory was unable to confirm, and that was one of the serious questions that I asked during the presentation. And I don't have a good answer because that's a risk decision that each manufacturer has to take based on their knowledge of their products, their process, and the methods that are being deployed by the laboratory. Next, please.
The next question is, do your testing methods accommodate the results of high pressure processing where inactivated or killed organisms are present?
Okay. I think the question is related to the viable but not culturable, or the not culturable because they're dead dilemma. So high pressure processing, obviously if you have a large population size of pathogen in the product, which I would say is probably very, very uncommon, because why would you be using raw materials and ingredients that have such a high bolus of microbes in that product prior to processing? So I would say that, again, that's not a usual situation. But let's say you do, and you apply a kill step. It could be high pressure, it could be heating, it could be whatever. Then the detection platform has the potential then to detect large population sizes of the pathogen, whether it's antibody-antigen reaction or a PCR reaction. And so most of these detection platforms, you actually have to cultivate and grow those microbes to get to a detection level. And if the residual amounts are below that detection level, the platform is hopefully not so sensitive it will detect those and give you a presumptive positive, even though you've got dead cells. I know that was a long, rambling answer, but we don't find that very often, where you start with high populations in your specimen, you kill those, and then you challenge your detection platform and they come up positive, even though the cells are dead. I hear that's a criticism of the methods, but we just don't see that very often. Next, please.
Okay, the next question is, are the facilities required to report to FDA when there is a positive salmonella? And then, and for customers who have a kill step and who will be further processing the product?
Okay. I'm not sure. I'm going to answer this question in two ways because I'm not sure from what direction the facility is referred to. So I'll start by talking about the laboratory. If the laboratory finds a test result that is positive, whether presumptive or confirmed, then that analytical piece of information goes to the customer. So the laboratory is not required to report those test results to a regulatory body. And the reason why is very simple. We really have no control over the customer products. We have no control over how the sample was obtained, how it was shipped, and so forth. Furthermore, we don't know if the product is actually finished product, in-process product. We could have a customer that's doing a process validation, so half the samples they send us are going to be positive, and after processing, half the samples should be negative. And so again, expecting the laboratory to report that to the regulatory agency without having knowledge of how the sample was collected is unreasonable. The second way I'll answer this question, if you are a producer, a processor, that is manufacturing a material or ingredient that there will be a subsequent kill step applied by your customer, then no, you're not required to report that to the reportable food registry. However, with FSMA, what you will be expected to do is to get a letter from that customer stating that they are okay with receiving product that has pathogen and that they will apply a subsequent kill step before releasing that product to the consumer. So you really need to be sure that you understand what the intended use of your product is down the chain of custody of that product by your other subsequent users. Okay, next please.
Next question is, do you recommend use of ELISA kits to test for peanut allergens, or do you recommend a DNA test?
Okay. It really depends on the nature of the specimen. For example, if you've got very low levels of allergenic protein in the specimen, and that allergenic protein is below the limited detection of the ELISA kit that you're doing, excuse me, obviously you'll get a false negative test result. And there are many circumstances, depending on the allergen of interest, where that might occur. Let's say the allergen is complexed in a complex food matrix. The allergenic protein could be denatured, making it less responsive to the antibodies in the ELISA kit and so forth. Under those sorts of circumstances, then one might want to do a PCR assay for the presence of peanut DNA as a way to see if your ELISA kit really has the kind of sensitivity that you desire. So what you're doing with a PCR test, instead of measuring the allergenic protein like an ELISA kit, what you're looking for is the source, possible presence of the source material. In this case, it could be something like peanut DNA or bovine DNA and so forth. Next, please.
Okay, the last question in the queue right now is, when a customer does not require for us to do a micro testing, are we required to notify the customer of results when we do routine testing for micro that they do not require?
Okay, I'm going to repeat the question just so that I kind of understand where it's coming from. So if you have a customer that does not require micro tests, but you get a positive pathogen test result, are you required to inform the customer of that result? Well, I would say you've got a very significant moral and ethical dilemma if you do not, because basically what you're doing is, I'm assuming your customer expects to receive product from you that does not contain pathogen. And if you have reason to believe through your test result, for example, that lot that you just shipped has pathogen in it, then I think you do have an obligation to inform that customer of that test result.
Okay, that was our last question in the queue for today. Sorry, we just got one more.
Okay.
This question was, what would be the recommended size samples if finished product is apple juice, concentrate, bulk? Says lots size 50 containers per lot.
Okay. I can't answer that question because I don't know what the questioner wants to test for. But you could do, again, an analytical unit, according to most people in the business, is going to be 25 grams. And so if you're doing a pathogen screen, you could do a 25-gram sample. Obviously, if you increase your sample size, you're going to increase the odds you're going to find the pathogen of interest. Another way you could do this is, instead of doing one large enrichment, you could do multiple small enrichments. But that's going to increase your testing costs. So if this individual who asked this question can reach out to us by email, maybe we can get further details to be able to give a better answer than the one I just gave.
He did just reply, and he said that this person said that they're testing for E. coli. And it says, yeah, E. coli.
Okay. Well, remember, the laboratory is only going to express results per gram. So most of the E. coli methods that we're going to use, we're going to require about a 25-gram sample. So that would be my recommendation just for E. coli.
Okay. This is going to be our last question due to time. Do you retest an enrichment if you have a positive result?
Do we retest enrichments if we have a positive result? Only on the request of the customer, because that's going to add cost to the tests. And if we're only reporting a single test result, then if a customer wants an additional retest of that enrichment, we're happy to do that. But that's not routine in our laboratories.
Okay. Thank you very much, Dr. Marshall, for your time today. Also, thank you to everyone who listened to the webinar today. Everyone will be getting an email follow-up tomorrow with a copy of the webinar and the slides. Again, I'd like to thank Dr. Marshall for his time. Everyone, have a great rest of your day.
Thank you very much. Bye-bye.